So thank you, CI Group, once again for a really nice presentation. And we have a time for a few questions right now. If anyone would like to start. Not, I actually have several. So.
Oh, okay, go ahead, start. Go ahead. It's a question more out of curiosity, since you provide these fantastic resources and you already said how much effort you make to make it accessible to the biologists. I wonder how much interaction you really get back. Like, what do you know about the users of your resources, and do they frequently communicate with you?
Well, yeah, we get a lot of questions. Also, last year, the last 2 years, we've given 26 workshops on 4 different continents. So we've been moving out a lot, trying to get people to use it. The main problem is that a lot of people are wed to their specific resource that they're used to using, because biologists, you can really, and I'm one myself, I like to do what I know how to do. I don't want to learn anything else. Even if you tell me it's 2 seconds faster, I don't care, because I don't have to experience a learning curve. So it's only now, I think, now on the site we're adding genome assembly and annotation, and these are then taking that data and downloading it into the resource. So I think once we start offering things that no other resource has, we start getting more. But the user questions are really going up. We're getting cited a lot more. So I do think we're making some inroad, but it's just hard because people have like Tuberculist, but that they all use. It's the simplest website in the world. I use it too because it is so simple, but it doesn't have the same power.
And just to dive on that also is for the RNA-seq website, what we found is, or I've been amazed by the level of detail people have sent in their email. They've sort of described their entire experimental setup, you know, their controls and what the whole thing is for, and then the problem that they're having. And so, you know, in terms of that, it's been really interesting to see the wide variety, or people requesting new genomes. You know, hey, I want to work on zebrafish for some disease model. Can you put that genome on your website? And so it's really great to. And then you have people thank you. Oh, you know, I can't believe I would have never gotten through this. And so that part has been really nice as well.
In a computational institute, I don't think you understand the problems that biologists have. We have had people send us their hard drives that they got from their sequencing center because they don't even know how to find the file that has the results. It's that, I'm serious. It's really hard for them. So we try to make it easier for them.
Yeah, we have some of those folks. They're going to be extinguished pretty soon. In your atherosclerosis study, I wonder, so the samples that you use, are these human samples?
Yes, actually we use the old PDA sample where it's true clinical sample that we got 1,100 PDA samples. We also have the de novo samples that they're producing right now.
What is the...
They got them from car accidents, people who had died, and for years they've been collecting these things for people who had, also people who had heart disease, but they would collect these, and some of them were in formalin fixed. So those have presented problems for the proteomics.
Yeah, exactly.
So yes, they are human samples taken from upper aorta, abdominal aorta.
And it's the same for your normal control samples for the comparison, coming from the same
yes,
group.
from the accident victims.
How do you know when, sorry, my ignorance on this, how do you know when a mutation is directly related to atherosclerosis or as a polymorphism of the gene?
They do have genome-wide association studies where they took over 20,000 samples that from those samples that they can associate those CAD cases that are linked to those specific loci.
Okay, thank you.
So Even though we're running a little bit behind, I get to ask my question because I'm the moderator, right? And so, Andrew, I just have a quick question. This RNA rocket, I think, is a wonderful resource that you're developing, but how closely is it integrated with the rest of the parts of the CI group? Because, you know, we're getting into this stage where it's not just transcriptomics, but it's meta-transcriptomics, right? And so the processing capabilities with the GTF files are going to be growing exponentially. So do you guys have a way to get around that, or what's...
Right. So it's being there's two answers to your question. One is the site itself is a byproduct of this entity called Pathogen Portal, which is over all of the bioinformatics resource centers under NIAID. So there's vectors, viruses, bacteria, and eukaryotic pathogens. So it includes like fungi, DB, and things under the UPath rubric as well. So but then once you get your results, the result files, you can download them and take them away, or you can just upload your results and do all your analysis online and even stream the results back to the respective BRC that provided the data in the first place and view it on their genome browser and everything like that. And in terms of computation, we host the site on a cluster in VBI. So at one point we did move it up to Amazon Web Services, and we found that the cost model wasn't, perfectly attuned to the grant cycles. It was somewhat of a challenge there, and sort of you pay as you go. Whereas in VBI, it provides some insulation for a fixed cost solution. So it is hosted on our own cluster here, which is nicely maintained by the RT group here. So.
Great. Well, if we can thank the CI division once again for their nice presentation.